Welcome back to eCoreAcademy, everyone. My name is Anita Raj, and today, as you can see, I'm bringing to you all a new Biology 1 episode. This series is still pretty relatively new on our channel, and if you haven't watched the previous videos, I would suggest you do that. My last video, which is the process of science, part 3 of 3, is going to be linked in the description box, so I would suggest you check that out if you haven't already. If you haven't seen any of the videos in this course that we're starting, I would suggest you go to our actual channel, hit the playlist button, and then you'll see Biology 1, and I have all of my videos organized there for you guys to check out. So I'd suggest you do that first before watching this video. If you've gotten to this video, well, congratulations, you stuck with me so far. I know the scientific method can be pretty tedious, but now this is kind of a transitional video between more of the concept of coming up with experiments, and then we're going to be transitioning more into aspects of life, like biotic things, pretty much what biology is based off of. This is going to be our last video before we delve into more organic chemistry, which is the biological macromolecules that I was talking about and referencing in the past few videos. So today we're going to be talking about characteristics of life. So in this video, I've split the topic into two parts. The first half of this video is going to be pretty much a review of some concepts of the scientific method. So I'm just going to be giving you scenarios. I have 3 scenarios, and then we're going to be breaking them down. For example, we're going to be identifying what the problem is based off of the scenario. We're going to come up with a hypothesis. Then we're going to identify dependent, independent variables, and then come up with an experimental and control group. So we're pretty much going to be dissecting a scenario, and the scenario is only going to be summarized in one sentence. So we have to derive all this information from that one sentence. I'm going to show you how to do that because you're going to be expected to know how to do that. Then we're going to get into characteristics of life. So we want to understand what constitutes life. So what characteristics does just matter have to be classified as living versus non-living? So I'm going to be showing you nine characteristics that something has to have for us to consider it a form of life. I'm going to be defining these different characteristics, and then I'll be giving you a good amount of examples. So don't worry if you don't understand it just by the definition. I'll give you plenty of examples, and I'll try to explain them to the best of my ability. Before I get into this video, I'd really appreciate it if you hit the like button, hit the subscribe button, hit the post notification bell, drop a comment down below, and share this video using our socials. Now let's get into the video. Like I said, the first part of the video is going to be a review of the scientific method that we've been learning in the past 3 videos. This is pretty much how should you break down these scenarios and transform them into experiments. Like I said, I'm going to be summarizing a scenario in one sentence to all of you, and these are pretty common scenarios. For example, the scenario might look something like electrolytes enhance sports performance. That's just an example of a scenario summarized in one sentence. Based off of that scenario, we have to come up with a question that we can base our whole experiment off of, and I called it the problem. So what is the problem here? Well, we don't know if electrolytes enhance sports performance. So our question would be, do electrolytes enhance sports performance? Then we'll form a hypothesis, determine our independent and dependent variables, define an experimental group, and define a control group. So that's pretty much what we're going to be doing on the next 3 slides. This is situation number 1, and as you can see, I put pictures here because I know some of you are visual learners. In this first situation, we have a person holding a cigarette in their hand. And the scenario is cigarette smoking increases the risk of lung cancer. So this is just a statement we're making. And now we have to form a whole experiment based off of this one statement because we want to see if cigarette smoking actually does cause lung cancer or if it doesn't. So here is how you would answer the following components of the scientific method. First, you have to identify the problem, and what I would do is I'd actually frame it as a question. So it's like you're asking a question, and you're going to base your whole experiment off of this question like we talked about. Remember, the first part of the scientific method is making observations. So you're observing the world around you and natural phenomena. So, for example, maybe you observe that maybe you're a doctor, and then a lot of your patients who come in with lung cancer are former smokers. And maybe you're trying to form an experiment based to see if there's a relationship between smoking and lung cancer. So what you would do is, after collecting your observations, then you would form a question. So in this situation, the question, I've written it as the problem. So what is the problem we see? Well, the problem is that this question is unanswered. So we need to form a question and then use our experiment to answer it. So as you can see, the problem is up here. What you do is you pretty much rephrase the scenario. So does cigarette smoking increase the risk of lung cancer? So based off of this scenario, we're now forming a question. We're asking this question because now we have to answer it with either a yes or a no. Afterwards, you do your background research, meaning you just see common causes of lung cancer. Maybe you do more studies on the anatomy of the lung. Maybe you do more studies on what are the chemicals and components of typical cigarettes, just things like that. Afterwards, you want to form your hypothesis, which is an educated guess based off of background research and pretty much your opinion. So what you think is right. So do you think cigarette smoking causes lung cancer, or do you not think it causes lung cancer? In this case, I wrote the hypothesis as if someone smokes, then his or her risk of getting lung cancer increases. So this is pretty much saying, yes, if someone does smoke, then that smoking is correlated to the fact that they may get lung cancer in the future. So they're at a higher risk of getting lung cancer versus someone who doesn't smoke. And if you see, I don't know if you've noticed, but in the past few videos, every time I write out a hypothesis, I put it in the form if then. Usually you want to write your hypotheses this way. As you get into higher level sciences, your teachers might not specify exactly how they want you to write the hypothesis, but this is my go-to form. I always use the if-then form because in math, when you're doing proofs in geometry, you actually do write things in if-then form. So I would suggest you apply that same concept to science and play it safe and always write it as an if-then form because you never know if your teacher's already going to expect that. This is just the safest way to go. So remember, whenever you write a hypothesis, you're pretty much writing if I or someone do this, then this will happen. So if it comes with the first part, so if an action is done, the then part goes with this is the result. So that's how you write a hypothesis. So in this case, that's the hypothesis. Next, we want to identify the independent variable. So the independent variable, remember, is what we as the experimenter are manipulating. So we have control over this. We're changing it. We're manipulating. We're controlling it. Um, and and here in this case, it's the amount of cigarettes smoked per day. So in this experiment, we might get two groups of people. I'm just making this off the top of my head. We might get two groups of people, 10 people each. Group A is going to be smoking cigarettes, and Group B is not going to be smoking cigarettes. So in this case, we're controlling the amount of cigarettes they're smoking. Maybe we have every individual in Group A smoke, 10 cigarettes a day, but then we have the individuals in Group B smoke no cigarettes every day. So in that case, we're able to control how many cigarettes the individuals in each group, so meaning the test subjects, are smoking. So in this case, our independent variable, what we can change, is the amount of cigarettes the test subject smokes per day. The dependent variable, or the responding variable, this is what we cannot control. Remember, this is what is responding to the independent variable. So while we can control the independent variable, we cannot control the result of our actions. Think of it that way. We can perform these actions, but we can't control what happens in response. So the dependent variable is going to be the deteriorated health of the test subject. So after maybe our experiment lasts about 30 days per se, let's say, and then after that we measure the health of the test subject. So maybe we measure the efficiency of their lungs or something after that. Every individual in both groups. And then based off of that, we can come up with some kind of measurement. Remember, dependent variables are usually some kind of quantitative or numerical measurement. So like inches or pounds or kilograms, something like that. We'll come up with a numerical measurement, and then we'll determine the health of each test subject after the experiment, because the health of the test subject is going to purely reflect if they smoked cigarettes or did not smoke cigarettes, and then we can see if there's a relationship. Then let's identify the control group and the experimental group. Remember, the control group is the group that does not get the experimental factor. Remember, the experimental factor is the independent variable. So the control group is going to be treated as naturally as possible. They are used as a basis of comparison by the end of the experiment. So the control group is not going to change. You can think of them as the normal population of people who don't smoke cigarettes. And in this case, I wrote someone who does not smoke. So you're going to have those 10 individuals in Group B be individuals who are not going to touch any cigarettes. And the reason we have those individuals is because we have the experimental group. The experimental group, which is group A with those 10 individuals, are going to be the individuals who do smoke. So by the end of the experiment, after those 30 days, when we measure the health, maybe the lung efficiency of each test subject, obviously the people in the control group are going to exhibit pretty normal lung efficiencies, whereas we can base the lung efficiencies of the experimental group based off of the control group. So if the lung efficiencies of the experimental group are much, much lower than those of the control group, then we can draw the conclusion that, yeah, maybe cigarette smoking does increase risk of lung cancer. So hopefully that was helpful. That was situation number 1, and I know I took a little bit more time in explaining each term, each kind of component as we went through it. But in these next 2 examples, we'll pretty much fly through them because hopefully now you get the gist of how to break these one-sentence scenarios down into a full-fledged experiment. So this is situation number 2. As you can see in this picture above, I have this little breakfast burrito almost. If you don't live in America, you probably don't know what this is, but it's pretty much taking bread, and I think they put egg in this one, but putting egg inside, and here's some fruit off to the side. This is just what's typically eaten in the morning before going to school or work. So the scenario here is eating breakfast increases performance in school. So maybe I'm a teacher and maybe I'm observing my students in class, and I'm just seeing that maybe I put out a poll asking, do you eat breakfast in the morning or do you not eat breakfast in the morning? And let's say the kids who were a lot more attentive, a lot more talkative, a lot more engaged, and just doing better in school said that they ate breakfast in the morning. And let's say the kids who don't eat breakfast in the morning were a lot more energy deficient. They weren't as engaged. They just weren't performing as well in school as the other kids. So maybe based off of that observation I had, I formed the question, which is the problem here. Does eating breakfast before school increase performance? So I'm not sure. I've made some observations. I've collected some data. Remember, discovery science. I've collected some data. I've looked at it a bit, and I'm asking this question. I'm posing this question that I can build an experiment off of. Does eating breakfast affect your performance at all in school? So after that, I do some research. Maybe I research how much sleep a teenager needs, how much food intake a teenager needs, just things like that. And then after I do that research, I need to form my hypothesis. So based off my research, I form an educated best guess based off of my opinion. In this case, the hypothesis is an if-then statement. Oops, I forgot to put the then here, but pretend there's a then right before the he. So if a student eats breakfast in the morning, that's the action that this test subject is doing. So if the student eats breakfast in the morning, then he or she will perform better in school. So remember, the if part is the action that myself or the test subject is performing, and then the then part is going to be the result of that action that I am predicting. So I'm thinking that if a student does eat breakfast, then he or she will inherently do better in school. After that, when we start talking about forming the experiment, let's talk about the independent and dependent variables. Remember, independent variable is going to be the experimental factor. It's what we control and what we can change. So whether breakfast is eaten or not. So maybe I have a class of 30 students, and I split them into group A and group B. I have group B eat breakfast every single morning before coming to school, and maybe I have group A eat breakfast every morning before they come to school, and I have group B individuals not eat breakfast before coming to school. So in this case, I'm changing the fact of whether they eat breakfast or they don't eat breakfast. That's why the independent variable is whether breakfast is eaten or breakfast is not eaten. That is what I, as the teacher, am changing. The dependent variable is obviously the responding variable, so what I'm going to be measuring. So what is going to be responding to the fact of whether these kids eat breakfast or don't eat breakfast. In this case, maybe I give them a test first period on the day that they come to school, on the day that I have half of them eat breakfast and half of them not eat breakfast. In first period, as soon as they come to school, I give them a test. And then after that test, I grade those tests, and then I put them in 2 piles. The first pile is a pile of kids who did eat breakfast, and then the pile of tests of the kids who did not eat breakfast. And then I just compare those grades. Maybe I calculate the mean of those grades or something. And I calculate the mean, and then I compare them. So we can see which group just did better overall in the assessment. So that's the dependent variable. That's how I'm going to measure if eating breakfast does increase performance. The control group, remember, is the group that stays natural. So we really don't give it any experimental factor. In this case, the experimental factor is if the group is eating breakfast. So let's think of it this way. Naturally, let's just assume kids don't eat breakfast. So think of food as the experimental factor. If I'm giving you food, you're receiving the experimental factor. Because the control group is not getting experimental factor, they're not getting any food. So that's going to be like the group B kids I was talking about, the kids who don't get any food, come to school, take the assessment that day. However, the experimental group is going to be the group A kids. Those kids are going to be the kids who receive the experimental factor, which means they eat food before they come to school. They come to school and then they take the assessment. So that was situation 2. Let's look at situation 3 very quickly. Here you can see Hummingbird. I love hummingbirds. They're so pretty. So in this situation, maybe I'm a farmer and I like bird watching, and I see that the feeders I put on my trees in my orchards and stuff. I see that hummingbirds are attracted to the color red. Maybe I observe that because I see a lot more hummingbirds go and get the sugar water from the red feeders as opposed to my blue feeders or my green feeders or my clear feeders. So maybe I observe this. And then afterwards, what I'm going to do is I am going to come up with a question that I can base an experiment off of. So my question is going to be here, as you can see next to problem, are hummingbirds attracted to the color red? So because I see a lot of them gravitating towards the red feeder, maybe I'm asking the question, are they attracted to red? So based off this question, I can form an experiment. Next, before I even make a hypothesis, I'm going to do some background research. So maybe I study hummingbird eyesight. Maybe I study the proteins in their eyes, like if they have opsin proteins and things like that, and I see if they can observe specific colors in the spectrum. Just do background research on those things. After that, I might form a hypothesis, the if-then statement. So here, my opinion after doing all that research is yes, hummingbirds are attracted to the color red. So the way I'm going to phrase that in my hypothesis, my educated guess, because remember, I'm still not sure if they're attracted to red, but I am pretty sure. It's my gut feeling based off of the research I've done. So I word it as, if a feeder is the color red, so that's what I can control. I put out a red feeder, then the result will be that it'll attract more hummingbirds. Next, if you look at the actual experimental part of this scenario, so the first part we're going to identify as a dependent variable. So this is what we are going to be changing. This is the color of the feeder. So we might have I'm going to say two types of feeders I put out in my orchard. The first is group A. Maybe I have 10 of these feeders and all of them are red because that's the color I think hummingbirds are attracted to. And group B, another 10 feeders are clear. They have no color on them. You can see the sugar water inside of them. They're clear. So in this case, the color of the feeder is what I'm changing. I pick either a red feeder or a clear feeder, and that is the independent variable. The dependent variable, what I measure, is the number of hummingbirds attracted. So maybe I don't have anything to do that day and I just sit outside, and maybe I count how many hummingbirds go to the red feeders and then how many hummingbirds go to the clear feeders. That will be my data. That will be my form of measurement. Then we'll identify our control and experimental groups. The control group, remember, is not getting any of the experimental factor. In this case, think of the red feeder as the experimental factor. Therefore, the control group is going to be the group of hummingbirds that go to the plain or clear filter. Meanwhile, the experimental group are going to be the hummingbirds who go to the red feeders that I put out, because the red feeders, remember, are the experimental factor. So that is situation number 3. We are done with the review of the scientific method, and now we're going to be moving into the second part of the video, which are characteristics of life. So in this section, I'm going to be going over 9 characteristics that typically symbolize that something is a form of life. And I want you to keep in mind as I go through this, what constitutes what is living and what is not. So as scientists, because remember, all of us are scientists in some way. So by applying these characteristics I'm going to show you, I want you to think, how do I determine if this thing is living or not living? These concepts are going to come in handy all throughout biology, because even though biology is the study of life, you really are going to you're going to encounter things that are not living. So I want you to keep that in mind. But the first 3 concepts are on this slide. The very first concept is called order. It means all things are made of cells. So think of it this way: cells are our smallest form of life. They're so microscopically small. You need to use like an electron microscope to see the organelles inside your cells. They think of them as the basic unit of life. Order is pretty much how we build off of a cell. We eventually get to populations and ecosystems. So we go from a cell, and multiple cells together make a tissue, and many tissues together make an organ, And many organs together make an organ system. And an organ system makes an organism. And an organism belongs to a population. And multiple populations in an area make an ecosystem. So that is like, I know that's a lot to take in, but if you're taking notes, I would replay that and I would note that down because that's very important. That's like literally order. That's the order of how you build from a cell to an ecosystem. So that is what I mean by order. All living things are made of cells. So I am made of cells. You are made of cells. Your dog is made of cells. Your cow is made of cells if you have one. Living things are made of cells, and hopefully the way I explained it, from a cell making a tissue to an organ system to an organism, we are all built of cells. Examples are cells that have specific functions make up tissues. Like I told you, your cardiac cells have specific functions. You actually have two kinds of cardiac cells, but we'll talk about that more in anatomy and physiology. They make up the electrical system of your heart. They might make up the chambers of your heart, the valves of your heart. You have other pancreatic cells, and you have liver cells, and you have stomach cells. So all of these different cells have different purposes. Your heart cells and your skeletal cells and your muscle cells, they're usually long and striated, meaning they help more with they have more endurance, and they have a higher concentration of mitochondria, meaning they have more energy, and they can just think of it this way. If you have muscles, your muscles help you with locomotion, meaning movement. So you have to have a lot of energy. They have to be powerful muscles. Whereas there are other kinds of cells that don't do those things. So cells have specialized purposes in your body. I also want to bring up the concept of a virus because I remember this confused a lot of kids when we were talking about biology. So a virus is not living because a virus is not made of cells. So you might be confused by that. Think of the perfect example is the coronavirus right now. So typically, viruses. I don't know if all of you know what they look like, but think of it like a spider. They have a really big head and they have like legs. What the virus is pretty much made of is protein. They're not made of cells. They're made of proteins. So the head part is called a capsid, and inside of the capsid is what is the RNA and DNA, whatever genetic code that virus uses. What that virus does is it attaches to a bacteria and it inserts itself and it inserts that genetic code into the bacteria so the virus can multiply inside of the bacteria. And then what happens is the bacteria explodes and produces more viruses. So the virus is made of proteins, not cells. So it's not a form of life. And lastly, cellular organization. What that pretty much means is exactly what I went over. Cellular organization is how we build from a cell to an organ system, organism, population, ecosystem. Cellular organization. Next, let's talk about regulation. So regulation, think of it this way. Regulation is trying to maintain something. Maybe you're trying to regulate the rules. So you're trying to maintain the rules. Maybe that's the best example I can think of outside of science. Regulation in this case is called homeostasis in science. It's the ability to maintain an internal balance. For all you chemistry people out there, this might be thought of as equilibrium. So what happens is if you have two compartments. So I have compartment A and compartment B, and I have a barrier right between them. But the molecules in both compartments can pass through the barrier as they wish. And I have all the molecules on this side and none of them on this side. Well, according to the laws of entropy and equilibrium, those particles are going to want to go into the other container and just spread out and just diffuse as evenly as possible because they just want to take up all the space. They want to be as even as possible. That's just the law of equilibrium. Things flow from areas of high concentration to high concentration to low concentration. They just want to fill up space. So I'm trying to use this as an analogy to this and the ability to maintain internal balance. Our bodies are always trying to maintain balance, and here are some really good examples I have. Regulation in blood flow through the vessels in large dog ears. Okay, so this is a really good example. When it gets cold outside, your veins and arteries will start to contract. So they will start to get a bit thinner because what happens is your blood is trying to stay warm because your blood is coming from your chest, which is perhaps the warmest part of your body. And our bodies maintain a very small range of temperature, which is considered suitable for ourselves from 98.6 degrees Fahrenheit. I don't know what that is Celsius off the top of my head. And what happens is if you're cold for a very long time, you might feel your fingers and your toes getting numb because these are your appendages. They're the farthest away from your heart. So it takes more to get the blood to your fingers because remember your veins and arteries are getting thinner and thinner and thinner as it gets colder. So what your body will try to do, in this case we use an example of the dog with really large ears, what your body will try to do is it'll try to regulate blood flow during the cold months especially during the cold months. It's going to do this during the warm months as well, but I think the best example is if you think of the cold months, it'll try to push blood to the dog's ears to warm its ears, warm its skull. So it's trying to maintain homeostasis, trying to maintain a 98.6 degree Fahrenheit or around that temperature throughout the body. So I think that's a great example of homeostasis. Another one is regulation of blood sugar levels. So blood sugar scientifically is glucose. So whenever you hear blood sugar, it is glucose. So I think this is super simple and straightforward. Your body wants to maintain the right amount of glucose in your blood, because what happens is glucose is used in cellular respiration. Cellular respiration is pretty much the production of ATP, which is an energy molecule. Don't worry if you don't know what I'm talking about. We'll learn all of this later on. It's a type of energy molecule that your body uses to perform the functions it needs to do. So regulation of glucose in your blood. And lastly, a fish maintaining water balance. I actually had to do some research before filming this video so I could explain this to you all properly. So think of it this way. A lot of fish live in seawater, and it's a very aqueous environment. And what they do is they actually intake a lot of that seawater. But just like us, there are molecules in that seawater that don't work for them, that their body just don't need or cannot use, like chlorine. So what happens is, to maintain water balance, a fish will use its gills. And what happens is it will get rid of the nitrogenous waste, so any waste that has nitrogen in it, it'll actually secrete it as ammonia and actually get rid of it through its gills. So ammonia is a polyatomic ion. If you don't know what that is, don't worry. That'll be covered in chemistry. But their gills actually turn this nitrogenous waste into ammonia that it can excrete through the gills and into the environment and get rid of that waste to maintain equilibrium, because you don't want that waste building up in the fish's body. Additionally, with the chlorine in the any kind of chlorine, the fish actually have chloride cells in their gills that maintain chlorine balance. So I thought that was pretty neat, and that does have to do with homeostasis, because the body just trying to maintain that balance. Next, we have growth and development. So grow and develop in a mature adult form. I think this is pretty straightforward. Think of a rock. So if you have if you take a rock and you place it on your table, and 20 on your maybe a table or your nightstand in your bedroom, and 20 years from now, if you look at that rock, it hasn't changed at all. It hasn't grown or just developed in any way. However, you in those 20 years that rock was sitting on your desk, you've changed so much. You've grown and you've just become a more mature adult. You've gone through, if you're a teenager, if you're a preteen or a teenager, you've gone through puberty. Your body has gone through changes. If you're a child, that's a big change as well. Hormones are being released. Your limbs are growing longer. Your brain is changing. The folds in your brain are changing. It's just all these different things that constantly are evolving in our body as we grow and develop into our mature adult form. So examples include the increase in height, such as the growth of an organism. So I think this is such a straightforward one. Think of how much you've grown in the past few years. Like that is indicative of the fact that you are living, that you're an organism, that you're growing. Because what happens is this goes right back to cells. You have cells, and these cells are dividing, and as they divide, you actually grow. And I think, honestly, the growth of any organism, maybe the growth of the plant in your garden, the growth of your dog, honestly, anything. And next is butterfly life cycle developmental stages. Same with the frog. Same with how the frog develops. How these organisms go through metamorphosis and they mature into adults. So this butterfly goes from being a larva, and its body undergoes these biological processes, and its cells will multiply. Different hormones will be released in the body. It becomes a pupa, and then eventually it emerges as its mature adult form. And what's very important is the mature adult form can reproduce, meaning it is gotten to a point where the body's fully developed that it can create another organism just like itself. So that is another indicator of life. So I'll go through these pretty quickly. The next one is energy processing. So metabolism and energy, and an example is plants obtain energy from the sun. So energy processing is, like it says, metabolism. So metabolism is how your body uses energy, like I said, ATP, how it converts things like glucose into ATP, which is usable energy to execute those biological processes. There are two main types of metabolism. The first is catabolism. It is pretty much the breakdown of molecules in your body. So, for example, when we eat things like plants plants actually produce glucose. And when we eat our body needs glucose. So when we consume but the thing is, when we consume glucose just is not a ready form of energy. And if you don't understand what that means, that's fine. We'll be covering that in a later unit. But our body just cannot readily use glucose. We need to convert it into something else that we can actually use. So what happens is glucose is degraded. So it's degraded, meaning it's broken down into pieces. It's broken down and eventually becomes ATP, which our body can use. On the other side, we have synthesis. So synthesis is building molecules up. Body also does that, and that's called anabolism. An example is plants obtain energy from the sun. So the sun, in the plant's case, all of you probably know about photosynthesis, but I doubt many of you know exactly how photosynthesis works all the way down to the biochemical level. And again, we'll be covering that in biology as well. So what happens is light hits, this is just a quick overview of it. Light hits the plant cell, and in the plant cell, electrons are agitated, and that agitation will actually produce energy for reactions to occur in the cell. So that is how plants obtain energy. So just the ability of an organism to obtain energy indicates that it is a form of life. Next, we have interdependence. So this is pretty much organisms relying upon one another. This has a lot to do with ecology. So when you study ecosystems and you look at populations of organisms, and maybe you see organisms within a population depending on one another, maybe a mother and a child depending on one another, or maybe you see organisms from different populations interacting and depending on one another in mutualism or commensalism, and we'll learn about all those things later on. I think the best examples that all of you are probably familiar with are consumers that obtain energy from plants. If you've all probably seen a very basic food web or food pyramid of ecosystems, and at the very I don't want to say the very bottom. At the very bottom, you have the producers, so your plants, your photosynthetic organisms. Then you have your primary consumer. Sorry, your herbivores. So like your primary consumers, and they eat those plants. And then you have the secondary consumers, and they'll eat the primary consumers, the herbivores. They're usually either omnivores or carnivores, and then you have the tertiary or maybe even quaternary consumer, and those will just eat the ones in the level below it. And then you'll have your decomposer off to the side. So the relationships between all of these organisms, this interdependence, because they each rely on each other. If you take one organism out of that pyramid is not going to, unless the organisms can adapt to the new ecosystem with that one organism removed, pyramid is going to collapse. So that's interdependence for you. And additionally, you see consumers that obtain energy from other organisms and harm those organisms, referred to as parasites. So you probably know about things like tapeworm. So those things will live in the bodies of other organisms, and they will just leach off of the nutrients and eventually just kill that organism. So I think that's another good example. So we have commensalism, mutualism, parasitism, and those are all the three main forms of interdependence. Next, we have heredity. So all living organisms contain DNA. So DNA stands for deoxyribonucleic acid. So it is pretty much what holds our genetic code. Think of it as that double helix. You've probably seen that like windy thing. I think I put it in my introduction video as well. It's just that windy double helix, and this contains all of our genetic information. It's pretty much the form of inheritance and heredity. And examples include DNA is copied before the cell divides, and the trait for hair color is inherited from both parents. So this all goes back to the cell. So remember how we said the cell is a symbol of life. So your cells have a nucleus, which is like the control center of the cell. We'll go over parts of the cell later. And then inside the nucleus, you have DNA stored. Your DNA actually contains the genetic code. It contains the instructions for what the cell should do. So it indicates when the cell should maybe produce a protein or do this or do that. It's instructions for what the cell should do, its purpose, and its function. So right before a cell divides, what it does is the DNA is copied under the process of mitosis, usually mitosis. And what happens is the DNA is duplicated. So I don't want to get into the technicalities of that, because if you don't know about mitosis yet, it might be a bit confusing. Just know the DNA is copied, because what happens is that one cell will split into two. And if we don't copy the DNA, those two daughter cells, the two new cells, that's what we call them, daughter cells, are going to have only half as much genetic information, half as much DNA as the parent cell. We don't want that to happen, so we duplicate the DNA So the new daughter cells have just as much genetic information and are properly functioning cells, because if a cell doesn't have the right amount of DNA, then it's not going to function properly. The other example I gave was the trait for hair color is inherited from both parents. So this is gonna be more about our topic with inheritance. So we do something we do a lot on DNA, which is like pretty much more structural based, and then we'll do more on inheritance, where we talk about how you get these traits. So we talk about the relationship between a parent and its offspring. And in this case, you have things called alleles. So for your hair color, I think you have two different alleles. You might have multiple, but let's just say you have two different alleles. And these alleles can determine everything, and one comes from each parent. These alleles can determine things such as your hair. I mean, in this case, I put hair color, but hair color, hair texture, just like your wave pattern, things like that. So those are all ultimately found in your genetic code. So your genetic code will tell your body what color to produce for your hair. Okay, so let's go to the last slide. The next characteristic of life is response to the environment. So response to external stimuli. So response to the environment means pretty much if something is happening in the environment, to be living, you have to respond to that. So if something stimulates your attention, stimulates some bodily process, you have to respond. For example, if I just put a rock, I don't know how to. I'm trying to think of the best way to explain this. If I like put a rock outside and it's shining super bright and it's like super bright outside, do you think that rock is going to complain, oh my god, it's so hot, or oh my god, I'm sweating, oh my god, I need water? No, the rock is not going to complain because it just can't respond to that stimuli. But we can look at examples of when we do respond to stimuli. For example, if you put a plant in a room and one side of the plant faces the window and the other side faces more like the darker part of the room, the plant is actually going to lean towards the sunlight because, as you know, plants need sunlight for photosynthesis. So it's actually responding to the environment. And you have another example. Fur coats in some animals living in colder climates are thicker and same color as environment. So I can give you a really good example. I love horseback riding. And the thing is, where I live, during the summers it gets hot, but during the winters it can get very cold. So in a lot of these stables, they have horses that just have not adapted to the environment here where we live. They just can't grow thick fur coats during the winter time. So those horses need to be sent down south where it's warmer. So, however, the other horses, they're able to respond to the environment. So when their bodies detect that change in temperature, they're immediately able to grow these fur coats and survive perfectly fine. In other ways, we have things like Arctic foxes, Arctic hares, and these animals actually have coats that match the environment that are white. Another example is a polar bear. That's just adapting to the environment. Next, we have reproduction. This goes a lot with growth and development, repopulating to make offspring. So examples include humans reproduce sexually, a cell divides asexually. So human cells, so in the male you have sperm cells, in the female you have egg cells. The egg cells so these kinds of cells, these sex cells, are considered gametes. And we're going to be learning about all this later on. I'm just going to give you a quick overview. Gametes have half as many chromosomes as the individual will have. These gametes combine to create offspring. So that's how humans produce, and they have to do it sexually because both a male and female need to be involved. Then a cell, however, divides asexually. So as humans, we actually are capable of reproducing sexually, but our cells themselves, like the cells in our body, reproduce asexually, meaning there's one parent. So one single cell splits into two. It's the parent to both of those cells. There's no female or male involved, just one individual. Plants also do this. Some plants also do this. Lastly, we have evolutionary adaptation. So populations evolve over many generations as individuals with traits best suited to their environment survive. So we'll talk about evolution. I actually finished it. I'm in AP Bio right now, but I just finished a unit on evolution. So evolution is pretty much changes in an individual. That's the easiest way I can put it. Adaptation is those changes, an individual changing so it's better suited to its environment, so has the highest rate of fitness and survival. In this case, populations evolve over many generations. You obviously all know that things don't change very quickly over time, especially when you're thinking about on the population scale. Things change very, very, very slowly over long periods of time. And individuals in these populations that have the best traits that are best favored to survive in these environments will survive. The reason this is a characteristic of life is because living forms are more adaptable than non-living forms. So as something that's living, I have cells in my body that can respond to stimuli in the environment and that can determine what they need to do now so that I as an organism have the best chance to survive and reproduce so I have offspring and my offspring carry on my genes and my traits. Some examples I can give you of this are the peppered moth evolution in industrialized England favored the selection of dark moths. So what this exactly means is that in England, after the Industrial Revolution, that means like a lot of factories were used, a lot of production. Smoke was given off by these factories, and in the forest nearby these factories, this black smoke, this like tar-like smoke, pretty much plastered against the tree trunks, and all the brown natural tree trunks literally turned black because of that smoke. And in those forests, there lived moth populations. And what happened is that the moth populations that were peppered, meaning they adapted to the environment, yes, because the tree color changed, they changed the coloration of their bodies. And the ones that were peppered, meaning they were both like either black or white, they had like a little bit of both. They were able to camouflage and blend into these newly changed trees so that their predators couldn't pick them off. So that their environment favored them. So that's what we mean by evolutionary adaptation. Those are the 9 characteristics of life. Overall, I hope you found this video useful and I hope you enjoyed the 2 parts. Obviously, I would suggest because they're very different parts, you go back and watch whatever you need and do whatever you need to. If you liked the video, I'd really appreciate it if you hit the like button, subscribe button, hit the post notification bell, drop a comment down below, and share it using our socials, Facebook, Twitter, and Instagram. Also, email us at eCoreAcademy@gmail.com if you want to discuss anything at all. Check out our website when you have a chance. Super useful, has so many functionalities that you should check out. That's all I have for you today. Please look out for my next biology video. Probably going to be on organic chemistry. Super excited to teach you all of that. And I hope you're all excited to learn that from me as well. All right, have a great day. Bye.